Hello, I'm Danny. I'm going to present you the project which I'm interested in, which this aims to express interferon alpha to test its interaction with measles virus accessory proteins. So just as a context, normally the body enters an antiviral state when it goes through an infection. And the antiviral state is a result of a single signaling pathway induced by interferon alpha. And measles virus inhibits host interferon signaling pathways, so the understanding of the molecular mechanism will help develop therapeutic strategies for unvaccinated children. So the project itself, we can express interferon alpha in E. coli and determine the expression levels using an ELISA his-tag assay after the interaction with measles virus accessory proteins. And the vector chosen for this project is the PR-set vector. This is a prokaryotic high-level expression plasmid with a strong T7 promoter, and it has a polyhistidine tag in the N-terminal, which helps in the purification and detection with antibody. So to insert our gene of interest, we use restriction enzymes and the available sites in the multiple cloning sites. And it should be noted that when a gene is amplified through PCR, restriction sites should be added to the ends of the genes. And then the vector can be digested and ligated to the gene of interest. And for the plasmid to get into the cell, we use top 10F cells. These cells are chemically competent cells for transformation, and moreover, the plasmid manual provided by Invitrogen has a detailed transformation protocol that can be used for this project. So very briefly, the protocol can be seen for practicity as three different stages. The first one is a treatment mainly with ice cold calcium chloride for cells that are about to be transformed. And afterwards, the transformation it's done by heat shocking the cells, and finally an incubation and the plating takes place. In the plating, the ampicillin resistance is taken into account, so ampicillin is added to the medium, and in that way we will make sure we only take selecting those containing the plasmid. And as a verification method after we transform our cells is diagnostic digests. This could be used roughly to confirm the plasmid structure based on the predicted sizes on the gel. And so the first thing we will need to do is extract the plasmid from E. coli, followed by the digestion, and then just run that into a gel. And this is how the results will look more or less. The first line is just a linearized plasmid, and the two different double digestions are to see if the insert is present, and finally a double digestion of the plasmid again, but this one should yield a linearized plasmid because the second site is not in the vector anymore, or it shouldn't be, because it was removed when the insertion of the gene was made. And alternatively, his-tag ELISA detection could be made to detect presence of protein, but that will rather be used for the testing interaction of interferon alpha with the virus proteins. Okay, and well, as I was saying, his-tag is widely applied for recombinant protein expression and binding assays to detect like protein and protein interactions. And the his-tag ELISA detection kit provided by GenScript will then help detect expression conditions and semi-quantitation of this his-tag protein, and in this case, interferon alpha. In that way, it can be achieved what was initially proposed and then find alternatives for unvaccinated children. Thank you very much.